Hello everyone, this is Sarita, Assistant Professor of Chemistry, Government Degree College for Women, Begumpet. Today let us discuss about chromatography. Basically, chromatography is one of the analytical techniques. It is mainly used for the separation of the components which are present in a given mixture. Today let us learn what is the main principle behind the chromatography, what are the different types of techniques, and what are the general terms that we use in chromatography. So as I said, chromatography is the separation technique So chromatography is a separation technique. Now, in this picture, you can see that if a mixture is given, we can separate all the individual components from the given mixture. Now, what is chromatography? How can we define this chromatography? So as I said, it is mainly a separation technique. So in this, we can not only separate the components, we can also analyze, identify, purify, as well as quantify the number of components present in the mixture. Now in this picture, you can see that this is the mixture, okay? So where in this we have 4 different components. So all these 4 different components can be separated from this mixture by using chromatography. So chromatography is a technique where it is used to analyze the components. That is how many components are present in the given mixture. Then we can also identify the components. That means whether it is an acid, base, or a neutral compound, that also we can identify. Then it is also used to purify. So after collecting or after separating the components, whether the component which is separated is pure or not, that also we can easily identify by this chromatography. And also it is used for quantifying the components. So quantifying meaning here is we can find out the amount of the component present in the mixture. So now in this, you can see the yellow component, which is larger in size, and the other components which are smaller than the yellow components. Now we can also identify the amount of the or the concentration of the components present in this mixture. So chromatography is mainly used to analyze, identify, purify, and quantify the components. Now this chromatography was first invented by the scientist Michael Sweat. So what he has done is he has done an experiment where he has taken the plant extract. This is the plant extract which is taken, and he poured this plant extract into a column. This is a column, okay. He poured this into the column where calcium carbonate is filled in it. Now here the calcium carbonate acts as the adsorbent. So as the plant extract is being poured into the calcium carbonate, what happens is the extract which is there in this, first it gets absorbed onto the calcium carbonate, which is acting as adsorbent, and then later on, after some time, the solvent gets percolated into the column, and then we can see a clear difference between the bands. All the colored bands are being separated from the solvent. That means by this experiment, what he came to know is this technique can be used for the separation of the components. Now, based on these color bands, he has identified the various components present in the extract, plant extract. So the various plant extracts such as xanthophylls, chlorophylls, and other colored compounds he has extracted in this way. So he was the first scientist to invent this technique. That is Michael Sweat. He is a botanist. In the year 1906, he has coined the term chromatography. So it is actually a Greek word where chroma means color and graphy is writing or study. So chromatography is nothing but we can study the components based on its color. But later on, other scientists developed this chromatography technique, and they have evolved the techniques, that is various techniques were evolved after that. Those are TLC, column chromatography, HPLC, GC, etc. So the scientists who have done more work on this technique were Martin and Sinch. So the full name is Archer John Potter Martin and Richard Lawrence Millington Sinch, or you can just simply call them as Martin and Sinch. So these two were the scientists who have developed this technique further, and they have won a Nobel Prize in Chemistry in the year 1952. So these two scientists, they have evolved all the other chromatographic techniques based on the same principle. Now, let us see what are the different terms which we come across in chromatography. The first term is chromatograph. So chromatograph, it is the equipment that enables a sophisticated separation. So as I said, there are different separation techniques. So each and individual chromatography technique should have some equipment. So that equipment is known as chromatograph. Then the other word is eluent. Eluent is nothing but the fluid which is entering the column, or it is the solvent that carries the analyte. Analyte means it is the sample which is having some, or the mixture which is containing the components. So the fluid, because we need to separate the components from the mixture, so we require some solvent. So that solvent is known as eluent. Eluate is another term, that is a mobile phase which is leaving the column. So what happens is when we add this mobile phase into the column from the top, so what happens is the mobile phase will interact with the sample, and then it, while traveling down, it will collect, or it will interact with the sample, and then it brings down some of the components along with it. So it comes out of the column. So when it comes out of the column, we call it as eluate. So eluent is nothing but it is the term used for the solvent, whereas eluate we use when the solvent, after running the, or after leaving the column, it is known as eluate. Then in all the chromatography techniques, we have two phases. One is stationary phase, the other is mobile phase. So first we'll see what is stationary phase. So stationary phase is a immobilized phase. Okay, so it is, that means it doesn't move. It is an immobilized phase. So it is immobilized on the support particles or on the inner wall of the column. So you can have the stationary phases as solid or liquid. Suppose if it is a solid, you can directly put it in the column, or if it is a liquid, then you have to make it immobilized by supporting it onto a separate adsorbents. Example, we can take silica layer. Silica layer is an adsorbent which is used in thin layer chromatography. Then what is mobile phase? So mobile phase is nothing but which is moving. Okay, so it moves, and now that is the mobile phase. What we can have is the liquid or the gas. So gas or liquid can be used as the mobile phase. The mobile phase moves through the chromatography column where the sample interacts with the stationary phase, and then it is separated. So the mobile phases, as I told you, that it is nothing but a solvent where it can be used as the mobile phase. It may be either liquid or a gas. Then what is retention time? So retention time is nothing but the time taken for a particular analyte to pass through the system. So when a sample is loaded onto the stationary phase, and when a mobile phase is run down the column, so what happens is the mobile phase, when it is interacting with the sample, it interacts with one or two components, and then it comes along with that of the component. So the time taken for that component or for the sample to come out of the column is known as retention time. Then the other term is sample or the analyte. It is nothing but the sample which we have to, for which we have to do this chromatography technique. And solvent. Solvent, any substance which is capable of solubilizing the another substance, that is We are nothing but, if suppose if we have any solid sample, then we need some solvent to solubilize it. So that is nothing but a solvent. Then the other term is chromatogram. So chromatogram is nothing but the visual output of the chromatograph. So after doing the chromatography technique, we need to detect the components. So for that, we will be using detectors. So those detectors may be spectrophotometer or mass spectrophotometer. And those detectors will, that is, a graph is obtained from that, which is nothing but chromatogram. So it is nothing but a graph where on x-axis we will be having the retention time. On the y-axis, there will be concentration. So the number of peaks here in this picture, you can see there are 2 peaks. That means there are 2 different components present in the mixture. So number of peaks will give the information about the number of components present in the mixture. Suppose if we have 3 peaks, that means we can say that there are 3 different components present in the mixture. And the area that is this one, this part, this whole part which is under this peak will give the information about the concentration of the component. So how much amount of the component is present in the mixture, that can also be detected by chromatography. Then we'll see the classification of chromatography. So this classification is based on the techniques by chromatographic bed shape. That means how the stationary phase is being loaded. Suppose if it is loaded in the column, okay, then it is known as column chromatography. If it is sprayed on a plane, then it is planar chromatography. So under planar chromatography, we come across 2 different techniques. Those are paper chromatography and thin layer chromatography. So in paper chromatography and the TLC, that is thin layer chromatography, the adsorbent is sprayed on the plane. So it is a planar technique. Whereas in column chromatography, the adsorbent is filled in the column. So it is a column technique. And the techniques by physical state of mobile phase. So as I said, the mobile phase may be gas or liquid. Suppose if the mobile phase used is gas, then the technique we call it as gas chromatography. If it is liquid, then it is liquid chromatography. The other one is affinity chromatography. So affinity is here based on the affinity between the sample, the stationary phase. So under this, we have an example of supercritical fluid chromatography. So we'll study about all these techniques in detail in our later classes. Now this is another type of classification where it is based on the nature of the stationary phase and the mobile phase. So in this here, you can see that there are different techniques again. So thin layer, CC stands for column chromatography, then high performance liquid chromatography, which is HPLC. In all these cases, the stationary phase is solid and the mobile phase is liquid. So here, solid and liquid, that is stationary phase when it is solid and the mobile phase is liquid, are seen in all these 3 different types of chromatography. Then if the solid is a resin, which is that too when it is an ion exchange resin, where it helps in the exchange of the ions. That is, the principle involved in this is exchange of ions based on the stationary phase where resin is used, which is having some polar groups. Then the mobile phase is liquid. So this we can see in ion exchange chromatography technique. Then the other one is gas solid chromatography, where in the name itself it is stationary phase is solid and the mobile phase is gas. Then similarly we have this one which is gas liquid chromatography, where the stationary phase is liquid but the mobile phase is gas. So generally when we have mobile phases as gas, okay, we coin the term as just gas chromatography. So based on the stationary phase, if it is solid, then we call it as GSC, that is gas solid chromatography. And if the stationary phase is liquid, we name it as GLC, that is gas liquid chromatography. And the other type is this one, which is gel permeation or exclusion chromatography. Now in this case also the stationary phase we use a solid matrix. Okay, so in the matrix or the gel we use in this case, so where the separation takes place based on the size of the molecules. The size of the molecules, whether if it is larger, the larger particles will be eluted out first in this case. And the mobile phase is again a liquid state. So it is gel permeation. Then the other type is where both stationary phase and the mobile phases are liquid. So that means it comes under LLC, that is liquid chromatography. Example is, one of the example what we can take in this is paper chromatography. So in paper chromatography, what happens is the water which is present, as we know that the separation is taking place on the paper here. So paper is made of cellulose and we have small pores in it. So these pores, in these pores, we have small water molecules which are embedded in these pores. So that is why liquid is acting as the stationary phase and the mobile phase is again a liquid. So this is the general classification of chromatography. The next we'll see what is the principle involved in it. So the main principle involved in all the chromatography techniques is mainly it is adsorption phenomenon. So you might have heard the term adsorption. There is another term which is known as absorption. So first we'll see what is the difference between absorption and adsorption. So absorption, this is absorption first. Absorption is nothing but one substance will penetrate into the bulk of another substance. That means the substance will go inside the another substance, whereas adsorption is a purely surface phenomenon. So you have to remember this in chromatography technique, it is the adsorption process what is seen, and it is mainly a surface phenomenon where interaction takes place only on the surface of one substance. So now in this picture, you can see the difference clearly. So in this adsorption, you can see all the molecules where they are getting adhered or they are, that is the because of the interaction between the solvent molecules and the surface of the substance. It is only the phenomenon you can observe is only on the surface. So because of some van der Waals forces or hydrogen bondings, they go and stick onto the or they go and adsorb onto the surface. They are not penetrating into the substance. Then in the later case, that is absorption, where the molecules you can see they are penetrating into the substance. Okay, so the molecules are drawn into the bulk of the substance. So that is the difference between absorption and adsorption. So in chromatography, it is mainly adsorption technique which is seen. Then what is the how the components are being separated in this chromatography? So the main principle, as I said, it is adsorption technique. So all the components will have some differential affinities towards the adsorbent, that is the stationary phase. So depending upon the different affinities they show towards the stationary phase and the mobile phase, the separation of the components can take place. We are also called as partition coefficient. So mainly when the substance or the sample is adsorbed onto the stationary phase, and then when the mobile phase is run, the sample will get partitioned between the mobile phase as well as the stationary phase. Therefore, the partition is different for different components. So based on that partition of the differential partition of the components, the separation is taking place. So affinity in turn is dictated by two properties, that is adsorption and solubility. So we can explain the separation of different components in the mixture by these two properties, which are adsorption and solubility. So now in this diagram, you can see it is a column chromatography where the column is filled with the adsorbent and the sample is loaded. Okay, this is the sample. or the mixture which is having some components. There are three components which has to be which we need to separate. Now what we are doing is we are pouring the solvent. This solvent is acting as the mobile phase. Okay. So when the solvent is being poured into the column, so what is happening you can see. So here, after some time, you can see that the bands being separated, different color bands we can observe. So as we run down this mobile phase, it interacts with the sample, okay? And then as the solvent is, the mobile phase or solvent is being run down into the column from top to the bottom, it is interacting with these different components, and then it is the separation of the components is taking place. Now, so two properties we can explain here. One is adsorption and the solubility. So one is where you can see this component, okay, light yellow, green colored component has traveled to a more extent, larger extent. So why it has traveled to a larger extent is because it is more soluble in the mobile phase. So when mobile phase is interacted with that green component, because the green component is more soluble in the mobile phase, it is traveling all along with the mobile phase. Then comes the red one. Red component is having lesser affinity or lesser solubility towards the mobile phase. And what about this blue band? Now, this blue band is having more adsorption towards the stationary phase. So when the sample is loaded, what happens is as the blue one is having more adsorption or more affinity towards the stationary phase, it binds strongly to the stationary phase. So when it is strongly binded to the stationary phase, it doesn't move to a larger extent when the mobile phase is run down, because mobile phase cannot deabsorb. That is, when this blue component is getting adsorbed, now what does the mobile phase do is it has to deadsorb the components, so based on its solubility. So in this, out of these three components, green colored band is more soluble, okay, whereas the adsorption of this green component is less when compared to the blue band. So that is why blue band is traveling at a slower rate when compared to the other bands. And the first component that can be eluted out will be the green band, because it is having more affinity towards the mobile phase. So what we need to remember in this is, higher the adsorption to the stationary phase, the slower the molecule will move through the column. So in this case, the blue band or the blue component is having more adsorption towards the stationary phase, and hence it is moving very slowly. Then higher the solubility in the mobile phase, the faster the molecule will move through the column. So higher the solubility in mobile phase, that means out of these three components, which is having higher solubility in mobile phase, it is the green colored component. So as it is having more solubility in the mobile phase, it is moving faster to the column. So all this depends upon the polarity of the compounds. So polarity of the compounds dictate their affinity towards the stationary and the mobile phases. So based on the polarity of the components present in the mixture, their affinity is decided. So based on their affinity, if it is having more affinity towards stationary phase, it will move slow. And if it is having more affinity towards a mobile phase, it will move fast. Then what are the adsorbents that can be used in chromatography? So the major types of adsorbents we generally use are activated alumina. Alumina, it is Al2O3. Silica gel, activated carbon, molecular sieve carbon, molecular sieve zeolites, and polymeric adsorbents. These are the various adsorbents that can be used. And these adsorbents will have its own characteristic properties. That is, they will have its own porosity. That is the pore size, pore structure, and what is the nature of its adsorbing surface, whether it is acid polar, non-polar, in that way. So each of these adsorbents, they vary in its own properties. And generally the pore size may be from 2 nanometers to 50 nanometers. So when the pore size is less, then the adsorption will be, then the separation will be very effective. So how to select the adsorbents? So selection of the adsorbents, first, what are the criteria or the points we have to remember while selecting the adsorbent is, one is solubility of the compound. So whatever the compound is given for you to for the analysis, first we need to know the solubility of the compound, whether it is hydrophilic or lipophilic. The nature of the substance to be separated. We need to know the nature, whether it is acidic, basic, or amphoteric. So by knowing the nature of the substance, we can select the nature of the adsorbent that can be used for the separation. Then adsorbent particle size. So as I said, the particle size matters a lot. The particle size should be around 2 to 50 nanometers. Then adsorbent should not adhere to the glass. So glass plate is taken in the case of TLC, that is thin layer chromatography. So we have to see that adsorbent won't directly stick to the glass plate. We need to add some binders to that. So where gypsum will act as the binder. Then reactivity of the compound with the solvent or adsorbent. Then we have to check whether there is any chemical reaction between the solvent and the adsorbent. So the solvent and the adsorbent should not chemically react. That we have to take care while selecting the adsorbent as well as the mobile phases or the solvent. Then chemical reactivity of compounds with binders. So binders should not chemically react with the solvent or the adsorbents. So these points we have to remember while selecting the adsorbents. Then what are the mobile phases that can be used in chromatography? So as I said, mobile phases may be a liquid state or gaseous state. So what are the functions of this mobile phase is, it is acting as a solvent. That means, suppose if a sample is a solid, we need to dissolve that sample in a proper solvent. So that mobile phase can act as a solvent where we can dissolve the sample. It acts as a developing agent. So it is mainly used for developing the chromatogram. Just now we have seen in the column where the mobile phase is being run down and the separation is being takes place. That separation we call it as developing. It acts as an eluting agent. So when it comes out of the column, we call it as eluate. So when it interacts with the sample, the components get separated and it is acting as eluting agent where it brings down the components out of the column. And what are the different mobile phases that we can use in the chromatography? It is here, it is given in the order, increasing order of their polarity. So we generally start from non-polar solvents or non-polar mobile phases. Those are carbon tetrachloride, cyclohexane, acetone, benzene, toluene. Then we can increase the polarity. So we can use esters, we can use alcohols, and then water, which can act as the mobile phases. Now in this diagram, you can see thin layer chromatography as well as column chromatography. So this picture is given here to make you understand that what is the nature of the components which are present in the mixture and which component is being eluted out first or which is being traveled at a faster rate. So in thin layer chromatography, this is the origin point where we load the sample, and after running the experiment, we'll find 3 different spots. All these 3 are colored. It is labeled as A, B, C. So from the origin, the distance traveled by the B component is more when compared to A and C. So here we are measuring from the origin. The origin will be this one, this point. Okay. And whereas in column chromatography, we load the sample here. Okay, at the top of the column we load, and then the solvent is being run down from the top. So when the solvent is run down, as I said, it interacts with the sample, and then based on the solubility of the components, in the mobile phase, the separation is being taken place. Now in this case, the yellow component, that is B component, is traveling at a faster rate when compared to A and C. So in column, we check the distance traveled from the top, from the loading point is here. At the top, the loading point will be there. Whereas in TLC, the loading point is here, the bottom of the slide. Okay. So even in TLC, you can see B is traveled to a larger extent. And even in the column, we can see that the yellow color, that is the B component, has traveled to a larger extent. So what does this indicate is the B component is having lesser affinity towards the stationary phase, and it is having more affinity towards the mobile phase. So the components which are having more affinity towards the mobile phase will travel to a larger distance. Or you can remember in this way, that is the components that adhere more strongly to the stationary phase travel more slowly. Okay, the reverse one, that is in this, you can see the C component has traveled to a lesser extent. Why it has traveled to a lesser extent? Because it is having more affinity towards the stationary phase. Even in this column, you can see the C component has traveled to a lesser extent. Okay, so more the affinity towards the stationary phase, that component will travel at a lesser extent or at a lesser distance. Whereas the component which is having more affinity towards the mobile phase will travel at a faster rate. So that we have to remember in all the TLC techniques, all the chromatography techniques. Then the other term which is very important is RF value. Now RF stands for retardation factor, or it is retention factor. So what does this RF gives information about is, it is nothing but we get information about the migration of solute relative to the solvent front. So what does that mean is, as we have seen in the previous slide, that the solutes have been traveled to, the components have been traveled to a different extent. Okay. So after doing the experiment, we have to calculate the RF value of each and individual component. So how we can calculate is by using this formula. RF is equal to distance traveled by solute divided by distance traveled by the solvent front. Now what is solvent front is, it is nothing but the distance traveled by the solvent from the origin. So in the previous slide, if we see, now this is the TLC. Now this is the origin line, okay, where the samples have been loaded, okay, and after the experiment, we need to measure the distances. So this is solvent front here. That means the distance traveled by the solvent from the origin is taken as the solvent front. Now, to calculate RF, what we have to do is, we need to measure the distances. So here the distances have been measured. Component C has traveled 1 centimeter distance from the origin. Then component A has traveled 2 centimeters. Component B, it is written here 3 centimeters, and the solvent front is at 5 centimeters distance from the origin. Now based on that RF value, RF formula, so RF value of C will be, that is the component C will be equal to the distance traveled by the component C divided by the distance traveled by the solvent front. So what is the distance traveled by the component C? Okay, it is 1 centimeter distance it has traveled. So it will be 1 divided by 5. 5 is the distance traveled by the solvent front. So 1 by 5, it will come around to 0.2 is the value. Then similarly for the component A. So what is the distance traveled by the component A? It is 2 centimeters. So it will be 2 divided by 5. It will be 0.4. Then for B, so distance traveled by the component B is 3 centimeters. Okay, so here we have measured it is 3 centimeters. So 3 divided by the distance traveled by the solvent front is 5. So 3 by 5, which is 0.6. So this is how we have to calculate the RF values after after doing the chromatography experiment. Now this RF value cannot be unity. Okay, it should be less than unity. That means the RF value should always be less than 1. If it is 1, what is the meaning when it is equal to 1? That means the components and the solvent front have traveled the same distance. Suppose if the RF, that is the distance traveled by C, is also 5, then 5 by 5 will be equal to 1. That means it is, if RF is 1, we cannot separate the components. So separation of the components is not possible if we are getting RF equal to 1. So what we have to do is we have to select another solvent. Okay, as I said, the solvents can be, we have to select the solvents, that is the mobile phases, based on trial and error method. So first, as we start from non-polar solvents, suppose if you are taking N-hexane, so suppose if we do with N-hexane, if we are getting RF equal to 1, then we need to change the polarity of the solvent. So we need to take another solvent. Suppose if you are taking benzene or alcohol, like that, the RF value we can calculate easily. So we have to see that always the RF value should be less than 1. Then only the separation is possible. If it is equal to 1, then there won't be any separation, and we cannot, that is the experiment, we cannot take into further. That's a wrong result what we'll get. So we need to change the solvent. And here the solvents, what we select as a mobile phase, we can take a single solvent or a mixture of solvents also. Mixture of solvents also we can take, that is by taking polar and non-polar solvents and mixing it in different ratios. It may be 1:1, 1:2, in any ratio. So it is all trial method. So depending upon the RF values, we need to change the solvent composition also, such that we can easily separate the components and we can get better results. Now, what are the factors which affect the RF value? So as I said, RF is very important factor. Now there are some factors which affect the value of RF, that is solvent system and its composition. So just now I told you, solvent system, that is a mixture of solvents, can also be taken in different ratio and its composition may also vary. So that this, by changing the solvent system, the RF value of the component the RF value of the components can also change. And temperature. So temperature should also it should be considered as one of the factor, because the temperature, generally we do all the experiment at room temperature. So if we increase the temperature, the RF value may change. So temperature is also one of the factor which affects RF. Then the quality of the paper. So quality of the paper here, when we are talking about paper chromatography, the stationary phase, which is a paper, so the quality of that paper will also decide the RF. So what is the quality of the paper, whether it is actually the paper, generally what we take in paper chromatography is Wattman filter paper. So it depends upon the mesh size of the paper, the porosity of the paper also decides, so which affects the RF value. And the distance through which the solvent runs. So solvent will run at different distances. Now suppose if we are taking TLC and if we dip the slides into the adsorbents, what happens is adsorbent sometimes it will adhere onto the glass plate with it may not be uniform adherence. Sometimes it may be non-uniform adherence. So if it is non-uniform, then the solvent distance traveled by the solvent will vary there. If the adsorbent or the stationary phase is uniform on the glass plate, then the solvent will run and it will show some distance. So it depends upon the uniformity of the stationary phase that is being adhered onto the glass plate, where it changes the distance traveled by the solvent, which ultimately affects the RF value. Then the quality and the nature of solvents. So the same thing again, the quality of the solvents, whatever the solvents we are taking, they should be pure. So we have to ensure that the solvents are pure. If it is impure, then again it will affect the RF value. So these are the main important terms that you have to first go through while studying the chromatography topic. So what we have covered is what is the principle behind chromatography. It is mainly adsorption technique, and the various phases which are involved in chromatography, those are stationary phase and mobile phase. So depending upon the type of the phases, that is stationary phase and mobile phase, the classification of the chromatography or the techniques, various techniques of chromatography are different based on these two phases. Then we have seen why some components travel to a larger extent and why some components travel to a smaller extent. So that is based on its adsorption or affinity and also the solubility. And then we have seen the RF, that is retardation factor, which should be always less than 1. So these are some of the terms. And what is eluent, eluate. So all these terms you have to just go through once we get into this chromatography topic. So hope you have understood the introduction to chromatography. Then in our later classes, we will see the chromatography techniques one by one. Thank you. Thank you for watching.